Hey everyone, and welcome back to the channel. Have you ever misplaced your keys or forgotten someone's name right when you needed it? We usually think of forgetfulness as a bad thing, but what if I told you that a simple moment of forgetfulness changed the world forever? What if one of the biggest discoveries in human history didn't happen in a brilliant flash of genius, but because someone just forgot to do the dishes? It sounds crazy, right? But it's true. Our story begins in 1928 in a cluttered London laboratory. A scientist named Alexander Fleming was about to head off on a well-deserved vacation, but in his rush to leave, he overlooked a small yet crucial task: cleaning up a stack of dirty petri dishes he'd been using for his experiments. He left them piled on a bench and went on his way, probably not giving them a second thought. Little did he know that messy stack of glass and gelatin was about to become the site of a medical revolution. When he returned to his lab 2 weeks later, he noticed something strange. Something that would lead to the discovery of penicillin, the very first antibiotic, and usher in the age of modern medicine as we know it. So, who was this forgetful scientist? Alexander Fleming was a bacteriologist working at St. Mary's Hospital in London. At the time, he was deep in his research on Staphylococcus, a common and often nasty type of bacteria responsible for everything from boils to life-threatening infections. His lab was, by many accounts, famously untidy. He had cultures and experiments everywhere. Before he left for his holiday, he had been growing colonies of this staph bacteria on agar plates, those little petri dishes. Now, standard lab procedure would be to sterilize and clean everything before leaving for an extended period. But Fleming, well, he didn't. He just left them stacked up in a corner. When he returned, tanned and rested, he started sorting through the old cultures before discarding them. Most were just overgrown with bacteria, as you'd expect, but one dish caught his eye. It had been contaminated. A fuzzy blue-green mold, similar to what you might find on stale bread, was growing on one side of the plate. Now, for most researchers, a contaminated sample is a failure. It's a ruined experiment, fit only for the trash bin. You'd mutter in frustration, toss it out, and start over. But Fleming was different. He was known for his incredible powers of observation. Instead of throwing it away, he paused. He looked closer. He was intrigued by what he saw. There was something truly bizarre happening in that dish. It wasn't just that there was mold. It was what the mold was doing to the bacteria around it. He noticed that in the area immediately surrounding the mold colony, the bacteria had been completely wiped out. It was as if the fungus had created an invisible, bacteria-free barrier around itself. Further away from the mold, the staph colonies were growing just fine, but anywhere near it, they were dissolving and dying. He realized he wasn't looking at a contaminated experiment. He was looking at a microscopic war zone, and the mold was winning. This was the critical moment of realization. Fleming understood that this fungus wasn't just a random contaminant, it was producing some kind of substance, a mold juice as he initially called it, that was lethal to the bacteria. He took a sample of the mold and cultured it, allowing it to grow in a liquid broth. He then filtered out the mold itself, leaving behind the liquid it had grown in. When he applied this filtered liquid to other bacteria, he found it had the same incredible effect. It stopped them dead in their tracks. The mold was later identified as a rare strain of Penicillium notatum, and so he named the active substance it produced penicillin. What was even more remarkable was that while this penicillin was deadly to a wide range of harmful bacteria, it appeared to be completely harmless to human cells. Fleming had stumbled upon the world's first true antibiotic natural weapon that could selectively target and destroy infectious agents inside the body without harming the patient. He had found a key that could unlock the fight against infections that were, until then, often a death sentence. But a discovery, no matter how brilliant, is only the beginning of the story. Fleming had found the magic bullet, but he couldn't make enough of it. Extracting and purifying penicillin from the mold was incredibly difficult, and the substance itself was very unstable. For over a decade, his groundbreaking discovery remained little more than a laboratory curiosity. He published his findings, but the scientific community was slow to grasp its potential. It was one thing to see a clear zone in a petri dish. It was another thing entirely to turn that into a medicine you could give to a sick person. Penicillin was so difficult to produce in any significant quantity that it seemed destined to be a footnote in medical history. That is, until the world was plunged into the chaos of World War II. The war created a desperate, urgent need for a way to treat infected wounds, which were killing more soldiers than the battles themselves. This urgency finally gave penicillin the push it needed. A team of brilliant scientists at Oxford University, led by Howard Florey and Ernst Chain, picked up where Fleming had left off. They devised a new, complex method for producing and purifying penicillin. They experimented on mice, and then, in 1941, they treated their first human patient. The results were miraculous. With the backing of the British and American governments, the race was on to mass-produce this new wonder drug. Pharmaceutical companies dedicated massive resources to the problem. They found better strains of the mold, developed huge fermentation tanks, and refined the production process. By 1945, at the end of the war, penicillin was being produced on an industrial scale. It was saving thousands of soldiers' lives on the battlefield, treating infections and preventing amputations. And soon after the war, it became available to the public. For the first time, doctors had a powerful weapon against diseases like pneumonia, syphilis, gonorrhea, diphtheria, and scarlet fever. The impact was nothing short of revolutionary. Lifespans soared. Surgeries that were once impossibly risky became routine. A simple cut would no longer be a potential death sentence, and it all started in that messy lab. So let's reflect on that. A forgotten petri dish, a speck of stray mold that likely drifted in through an open window, and one incredibly curious scientist who, instead of cleaning up his mess, decided to ask, wait, what is that? Alexander Fleming, along with Florey and Chain, was awarded the Nobel Prize in Medicine in 1945 for his discovery. Penicillin didn't just revolutionize medicine, it fundamentally changed our relationship with the microbial world. It paved the way for the entire field of antibiotics, saving, by some estimates, over 200 million lives and counting. It's a powerful reminder that sometimes the greatest leaps forward don't come from meticulously planned experiments. They come from accidents, from mistakes, and from the curiosity to look at the unexpected and see not a problem, but a possibility. All because one fateful day someone didn't clean up their lab. Thank you so much for watching. What do you think is the most interesting accidental discovery? Let me know in the comments below. And if you enjoyed this story, don't forget to hit that like button and subscribe for more dives into the amazing history of science. We'll see you in the next one.